This is a precast covering eye physiology. So the eye is designed to process light energy. Light is known as electromagnetic energy, sometimes known as wave energy, because the light energy is in the middle of a larger spectrum of wave energy that includes ultraviolet and infrared light, and in the extreme includes X-rays and gamma rays on the one hand, microwaves and radio waves on the other. What is visible to us within this light spectrum is essentially the wavelengths of electromagnetic energy to which our photoreceptors are tuned or can respond. And in this particular case, we have basically four different kinds of photoreceptors. One is known as rods, whose sensitivity lies somewhat within the middle of the spectrum, but it is also relatively broad and does not discriminate greatly amongst different colors. Whereas cones are distinguished by having three different types, blue, green, and red, such that the blue cones are most responsive to short wavelengths at one end of the spectrum, and green cones more towards the middle of the spectrum, and red extending to the longer wavelengths of visible light. The three kinds of cones together essentially represent the full range of colors within the visible light spectrum. Now, when light passes through the lens, and then actually passes through the cornea as well, it is bent by this lens. And that means that the light is essentially focused onto particular parts of the retina. Remember, too, that part of what happens to light as it passes through a small aperture is that it is also inverted or flipped. So as represented in this drawing, where the yellow part of the image is in this lower left-hand corner, now that part on the sensory surface on the other side of the lens is presented in the upper right-hand corner. So light is refracted at the cornea, it's refracted at the entering edge of the lens, and it's refracted again at the back or lens where the light leaves. And the curvature and shape will change the way it is focused and change the way that it hits the retina. The other materials through which light passes, the aqueous humor and the vitreous humor, all have minimal impacts on the refraction of light. And so they are part of the path, to be sure, but they are not part of the actual refraction. And we also mentioned the neural layer of the retina, the parts of the retina that precede the arrival of light at the photoreceptors, as also affecting the passage of light. They certainly can scatter the light to some degree, but they too are not refracting the light in the way that the cornea and the lens do. Now, as we said, an image is inverted as it passes through the pupil and the lens. And in order to be able to see a distant object, which essentially means to be able to have the image of that, the light image of that object, to actually hit the retina with clarity, the lens needs to be flat. Another way of saying this more simply is that the lens is flattened when you need distant vision. And that is achieved because the ciliary muscle is relaxed, and so these zonule fibers or ligaments are pulling on the outside of the lens and maintaining it in a flattened position. So here is looking at it from the back. So here's the lens suspended, the center of the ciliary muscle with the zonual fibers, suspensory ligaments connecting the two. And in the normal relaxed position, then these fibers are pulling out and flattening the lens. But under parasympathetic control, when when you need to be able to focus on something up close, in order to have that image faithfully presented to the retina, requires that the lens become rounder, to ball up. And that happens when the ciliary muscle contracts, like the circular muscle of the iris of the eye. When the ciliary muscle contracts, it constricts, and that brings the inside edge closer to the center, and that relieves the tension of the ligaments on the edge of the lens and allows it to achieve a more natural, rounder shape, and that rounder shape refracts the light in a different way than the flat lens, and is able to take an image that's closer up and still present it faithfully to the retina. So this process of lens rounding is known as accommodation. That's necessary for close vision, or near vision, as it's sometimes called. Along with that is constriction of the pupil, because you get a more faithful presentation of the image of something nearby if the pupil is constricted. There also has to be convergence of the two eyes on something that comes close compared to looking at something far away. So this is sometimes known as an accommodation or near vision reflex. Now, you may have noticed lots of people from about 40 years on having to wear reading glasses. And that's because the lens loses its normal elasticity or flexibility to change shape in this way once that age is reached. And it becomes permanently flat, largely because the normal vision that we engage in tends to be distance vision rather than close vision. Even though some of us are reading a lot, still we are normally not looking at things up close. And so it gets, when it loses its elasticity, it's almost always likely to be into a permanently flat position, which means that the only way to be able to look at things up close is to add an additional lens in the light pathway that will make an accommodation, an adjustment, but in this case, artificially through the use of reading glasses. Everybody goes through this as an adult reaching this particular age. And it doesn't mean that some people choose not to use reading glasses, but they are not seeing as well as they could if they used them. Now, completely independently of this is the kind of refraction problems that we associate with being nearsighted or farsighted. These kinds of problems are associated with the length of the eyeball being either too long or too short, which means that the focal point, the focusing of the light has to be adjusted in such a way. In the case of the myopic eye, the light has to be thrown a little extra distance in order to reach the back of the eye. And in the case of a farsighted eye, the lens has to be found that can keep it shorter. And so that's the difference between a concave and a convex lens. You can see that in this particular drawing. So here's a myopic eye. Emmetropic is normal length. A myopic eye essentially is operating assuming a normal length. So the focus is presented in front of the retina, And so it takes a concave lens to be able to spread out the rays enough to be able to send it at a greater distance. This is the kind of lens that is thinner in the center and thicker at the edges. And you should be able to notice people's eyeglasses and be able to tell if they're nearsighted. By the same token, a short eye is hyperopic or suffers from farsightedness. In this particular case, the light rays have to be brought shorter to be able to accommodate the shorter eye. And this is accomplished with a convex lens, one that is thicker in the center and thinner at the outer edge. Certainly nearsightedness is much more common than farsightedness. But as I said, these are independent of the need for reading glasses, what we call presbyopia. Now, as we said, the retina has the photoreceptors of two flavors: rods which do not discriminate color, and cones that do. But not responding to color is balanced by the fact that rods respond very nicely to dim light. And so when you are attempting to look at something at night or in the dark, your rods will be the most likely to enable you to see something, but you might find that you're not able to perceive much detail about the color of anything in such dim light. Note, too, that the rods are located the most peripherally within the extent of the retina, which means that they are most responsible for peripheral vision, that is, picking up sensory information coming in from either side of the broad visual field that each eye is looking at. On the other hand, not only can cones respond to color, but they are the ones that are packed in at the macula and fovea. So they are used for central or focal vision. So they are centrally located in the retina, and they are the cells that are processing information from the focal point that you're looking at in your central vision. They do respond optimally in bright light as well. And so during the daytime under normal light, you are probably primarily using your cones rather than your rods. We have used the term transduction generally to refer to the way that sensory stimuli produce changes in sensory receptor cells. Phototransduction is merely the term that refers to how light affects photoreceptors specifically, and how it leads to a change in their membrane potentials that's known generally as graded potentials. So light energy is converted into the electrochemical energy of the nervous system. As I said, this is what occurs in photoreceptors, but that means in rods and cones. Now, this happens because the rods and cones contain a light-absorbing molecule known as retinal. And it combines with another kind of molecule called opsin to form what we call visual pigments, chemicals that can absorb light. It turns out that the retinol is made from vitamin A, which is the basis for your parents insisting that you eat your carrots because it's good for your vision. In fact, it's true. You do need sufficient amounts of vitamin A to be able to make the retinal that allows you to see, to allow phototransduction to occur in your rods and cones. Now, the basic story about a retinal is that it comes in two shapes, and light hitting the retinal changes the shape and that shape change is what causes the graded potential. The change, the graded potential that occurs, is a hyperpolarization. And as you may recall from talking about neurons, a hyperpolarization takes a membrane potential further away from threshold, so that tends to inhibit a cell. Now, without going into great detail, I just present this to give you a sense of what we mean by a shape change. Here's a molecule of retinal, and its normal structure involves a bend or crook in the long chain or arm of the molecule. This is known as cis-retinal, and when light hits this retinal, it straightens out the long chain or arm, and at that point we call it all trans retinal. When that happens, it breaks up its relationship with opsin and initiates the process of changing the membrane potential that results in phototransduction. So, in the dark, photoreceptors are actually normally quite active. They have a dark current, a movement of ions, and so they are depolarized, which means that they are releasing neurotransmitter at the synapse with the bipolar cells. But it turns out that neurotransmitter is inhibitory. It elicits IPSPs, inhibitory postsynaptic potentials, and that inhibits the bipolar cell, and so the bipolar cell doesn't go on to stimulate the ganglion cell, which means the ganglion cell is not excited, so it doesn't send action potentials onto the brain. So, in the dark, without light stimulation, we don't see light, is essentially what this means, which should not be surprising. On the other hand, when light hits the photoreceptors and causes this shape change in retinal, this actually hyperpolarizes the photoreceptor. And this is true of both rods and cones, which means that it inhibits the release of neurotransmitter. But now what we're doing is blocking the release of an inhibitory neurotransmitter. And so that actually disinhibits the bipolar cell. Without the presence of inhibition, it actually allows the bipolar cell to become excited or depolarized, and that in turn will allow it to release excitatory neurotransmitter, which will excite the ganglion cell and get the ganglion cell to send action potentials to the brain to register in the brain the fact that your eye was just stimulated by light. Now, this is just a way of essentially finishing the story by pointing out that in order for us to see, the light signal has to travel from the eyeball along the axons of the retinal ganglion cells. They initially travel in the optic nerve from each eye, and then ultimately reach to a area known as the visual thalamus, aka the lateral geniculate nucleus of the thalamus, and then there are neurons there that send that information on to the occipital lobe, which is where the visual cortex lies. All of that has been dissected out in this view of a human brain. Notice what's important about this illustration is to remind you that we have binocular vision, which simply means that the visual field associated with each eye, here's the visual field for the right eye. The right eye is illustrated on the left here. Here's the visual field for the left eye. And so there's an intermediate point that is overlapping from the two visual fields. And this is where we have binocular vision. And so most of the time when we are focusing on something, we are focusing with both eyes. We have what's called conjugate gaze or convergent gaze. The two eyes are able to focus on the same things. And so that information is necessarily combined and shared as it's sent up to the brain. Now, what's indicated here is that any particular thing that we're looking at essentially is inverted except for the part that we're focusing on. So the fixation point goes to the fovea macula in each case. But in the case of the right eye, more nasal part of the visual field is going to hit the temporal part of the retina, and more temporal parts of its visual field are going to hit the nasal part of the retina. So light will be inverted in the same way that we have seen in previous examples. So that affects how the information is conveyed up in the brain. Now, this is a drawing that comes out of the central nervous system material, looking at the optic nerve. And color-coded here is how light coming from the temporal parts of the right eye and the nasal part of the left eye both converge onto the right half of the brain. So the nasal fibers always cross in a structure called the optic chiasm. This is where the two optic nerves meet just underneath the hypothalamus of the brain, just above and in front of the pituitary gland. And then the temporal fibers stay on the same side. And so because of that, you have a joining of fibers that are carrying information about the same part of the visual field. So in other words, the temporal part of the right eye, nasal part of the left eye, are both being stimulated by light from the left half of the visual field. And that information converges onto the right half of the brain, the right lateral geniculate nucleus, and in turn conveyed to the right visual cortex. So left visual field goes to right brain, and in turn, information from the right visual field, essentially following the same rules. Temporal information hitting the nasal retina, nasal information hitting the temporal retina, and the information converging onto essentially the side of the brain opposite from that visual field. So right visual field going to left brain. And that's the end of this precast on eye physiology.